It's been posted on Google Video and on ICON itself, so I hope that that's an improvement. There have also been posted on ICON a sample exam, if you want to use that as a study aid, and 2 different versions of review PowerPoints, one that is text only and one added just this afternoon that also includes some figures. Tim, do you have a time and a place I can announce for the... Sorry, what? Ah, okay.
It's going to be as close to 5 o'clock as we can get it.
Okay, 5 on Thursday or Wednesday? Okay, we're aiming at 5 p.m. Wednesday for a review session, but the person who schedules the rooms is not available yet, so we're going to have that as soon as possible. Keep your mental calendar open there, and we will mail that out as soon as we have a firm time and place. Is that pretty much current? Okay. Any other kind of practical questions and so forth? Yes? I certainly can. Thank you for asking. Okay. Should be down there soon. Any other practical questions? Or impractical questions? Okay. Then let's make sure. Okay. So we have finished up with the metabolic pathways aspect of our course. That is, we've gone through the let's see, here we go. We've gone through the sort of the local tours of lipid metabolism, nitrogen metabolism, and so on. Why is this not opening on me? There we go. Lipid metabolism and nitrogen metabolism, the great electron transport things like oxidative phosphorylation and photosynthesis. And those are the materials that will be incorporated into the 3rd exam. The material I'm going to be talking about today, today and Wednesday, integration and metabolism, will partly be summarizing and reviewing things we've already talked about. So those things, of course, are useful to you in your putting thoughts together. But we're also going to be trying to take a higher order view of things and see how these different pathways work together overall for the welfare of the organism. And metabolism is integrated really in a wide variety of ways. And it's essential that that happen because, first of all, it could end up being extremely expensive. And organisms in general try not to waste things. And secondly, there's always got to be the right things at the right time. It's sort of the original just-in-time version of things. We're going to look a little bit about how compartmentation contributes to the regulation of metabolism. And compartmentation operates at every level, from down at the level of the cell all the way up through the organism itself. You could even regard ecology as an integration metabolism between organisms. We're going to talk a little bit about hormone mechanisms and how cells turn something that's outside the cell into a useful signal inside the cell. We'll talk a little bit about some specific hormones, insulin and glucagon and some others that have an extremely important role to play all the time. And then as an example of integration metabolism, we're going to look at one of the grand disorders of metabolism, which is diabetes. We'll see why we're going to focus on that particular disease. It's a disease of both fat, of glucose and fat, not just of glucose, and a very sobering case study it is indeed. But there's optimism. There's hope. I would never leave you without hope. We've already seen, actually, a surprising amount of compartmentation of metabolism. But we've seen it at the level of the cell. And I don't know if you've noticed this as we've gone along. I've been mentioning it as we've gone along, but sometimes seeing it all in one place makes it easier to remember these things or to see the pattern to them. So if we think of 3 major compartments for intermediary metabolism in the cell, you have the cytosol, the sort of protein and stuff-filled space between the nucleus and the membrane. And you've got the matrix of the mitochondrion, which is that very dense, full area that's inside the inner mitochondrial membrane. And then we have the membrane itself. In this case, it's the inner mitochondrial membrane, which is where all the exciting parts of metabolism happen for the mitochondria. We can see some patterns. First of all, it's surprising to see just how much metabolism depends on the membrane. Many of these things are transporters, but the electron transport chain of oxidative phosphorylation is also a component of that inner mitochondrial membrane. So one of the very key pathways we've looked at is at the interface between the two. Out in the cytosol, we can see some things that are synthetic. So gluconeogenesis is synthetic, fatty acid synthesis, nucleotide synthesis. Some amino acid synthesis is out there, but some forms of simple breakdown as well. So glycolysis is out there. Pentose phosphate is really amphibolic. It can build things and it can break them down. It contributes to nucleotides as well as to carbohydrates. And parts of the urea cycle are out there. Parts of amino acid degradation are out there. If we go back inside to the mitochondrial matrix, we have the citric acid cycle, which is the grand amphibolic cycle of all time, the common ending point for oxidizing fuels, a rich source of starting materials for a lot of different things. We have the beta oxidation of fatty acids. We have ketogenesis, which is sort of building in order to break down. You build up ketone bodies so you can burn them somewhere else. Some amino acid synthesis and degradation also occurs in the matrix. And then we have the rest of the urea cycle. So clearly, it's not all happening in one place, that it's possible to segregate these components so that the can have separate pools of intermediates or separate regulation depending on the exact needs of that cell at that time. We also see compartmentation and cooperation at the level of organs. So we have a number of important metabolic organs, the liver, because its focus is on metabolism, our muscles, because of the mass of them, to a lesser degree, the kidneys and the brain, the bones. And to give you an example of this kind of coordination, let's look at anaerobic exercise. During anaerobic exercise, the energy demands put on the muscles may go beyond what the blood supply can keep up with or the mitochondria can keep up with, burning things oxidatively. And if that happens, if we have glycolysis and can't completely burn that pyruvate through the citric acid cycle and oxidative phosphorylation, to keep the process going, that pyruvate will be turned into lactate to reconstitute the nucleotide, the NAD, that's going to be needed. The lactate leaves the muscle, and in the liver is regenerated into pyruvate, and at the expense of ATP can be made again into glucose. That glucose returns through the bloodstream back to the muscle. And so the net effect of this circulation here is that energy is moving from the liver to the muscle to compensate for that anaerobic activity. If the liver didn't help out like this, you could end up with sort of local poisoning effects from the consequences of anaerobic activity. And other conditions, so for instance, under extremely vigorous exercise or if you're fasting, muscles may actually need to dispose of nitrogen. And what are some of the circumstances where that might be? Well, if you're burning amino acids for fuel, you're going to end up with trying to dispose of that. And the pyruvate can accept a nitrogen from amino acids through transamination. That provides that amino acid backbone now as an alpha keto acid that could be used locally. The alanine can act to carry the nitrogen to the liver, and in the liver, the nitrogen is disposed of through the urea cycle that we looked at just a day or two ago. The pyruvate, again, through gluconeogenesis, can be regenerated into glucose and back to the liver or to the brain, and so on. And in this case, the cooperation is moving nitrogen from muscles to the liver. So we can see how pathways that we're familiar with as individual pieces, in fact, are working between different organs to get the whole job done. Now, in terms of coordinating all of this, there's only so much that can be done locally, given the fact that the different organs can do different parts of the job, and given the fact that in different overall conditions of feasting or fasting, we have different demands. How do we coordinate all those things? And the broad coordination is effected through the body through hormones. The Greek roots from hormones come from the words for I urge on. So hormones are molecules, are chemicals that are produced in one place and have a metabolic effect or a biochemical effect somewhere else. But bodies can't read. There has to be some molecular way for a molecule to generate a signal. And we call that overall process signal transduction. That is, a hormone itself does nothing. It carries information, and somehow that information has to be turned into something useful. And when we look at what we know about hormones, we see that there is a broad pattern of how these things work. So if we start at the outside of the cell, and here we're going to treat the hormone as a ligand. It's one of the things that can bind to the surface of the cell. There are other ligands at the cell surface, but hormones in general are going to have to be a ligand to something. And if we're going to talk about ones that act at the cell surface, there's going to be some kind of a receptor, some kind of a transmembrane protein that can bind the ligand on the outside and have an intracellular domain that's going to do something itself. Frequently, the binding of a ligand causes that receptor to change its conformation. So a little event on the outside causes a ripple till the inside is different. Whichever way it occurs, there's now some new activity due to this change in the receptor, and there will be some series of intracellular components. The most important aspect of this is the capacity for cascades. And so when we look at things that are engaged in signal transduction, by having more than one step, you can amplify at each step and get a larger and larger signal. So that if one ligand activates the one receptor molecule, but that one receptor molecule generates, say, 10 of this intracellular component, which in turn can make 10 of that. Well, now we have 100 of this, for example. And this, in turn, may make a small molecule called a second messenger that will affect its changes one way or another, or it may directly target another protein one way or the other. Second messengers are common in these cascades, although they're not universal. And another aspect that's always important, although it's not always focused on or sometimes even understood, is that if there's an on switch, there's an off switch. There's some way for the whole cascade to come to an end. Many times these targets, one way or another, will be protein kinases. Not always, but oftentimes, because a protein kinase is a good way to amplify things. Activate one protein kinase, it can phosphorylate 10, 100, 1,000 other things. When we think about protein kinases, there are a couple of major families. There are others as well, but there are a couple of major families. And there's one group called the tyrosine protein kinases, in which the side chain that's going to be phosphorylated is a tyrosine. And the other is the family that will target either a serine or a threonine. And if you look at these, it won't be either or. It'll either be a serine or a threonine. Something to notice about all three of these is that they're taking what are initially electrically neutral side chains and turning them into ones that are negatively charged. So the analogy I tend to think of is, the door is a big thing and a very small doorstop can stick it open. So one phosphate group can have the effect of changing the behavior or the conformation of the whole protein. I'd like to talk for a little bit about insulin, which is one of our first understood hormones and one of the ones with a huge impact on medical care and the well-being of Americans and people in general. One of the major jobs of insulin is to regulate which fuels are going to be used at a given time. And it's the work of this little bitty thing here. It's a small polypeptide hormone. So it's a protein, or the byproduct of a protein because it's processed lightly. And by its concentrations in the blood and by which cells it binds to, it can modulate which fuels are going to be used at which time. And this turns out to be important because if we look at different metabolic states, so we can look at after a meal, which is called postprandial, where you've had a meal and you're as flush as you're going to be with things. In the postprandial or just after eating state, probably 50% typically of your energy is going to come from carbohydrates. Another third of it's going to come from fatty acids, and then a smidge, a little more than a seventh, comes from amino acids. If you fast overnight, so let's say you have dinner at 6 and you're going to have breakfast at 7 the next morning. Before you eat that breakfast, your body has, in that intervening fast, that overnight fast, shifted which fuels it's using at the moment. So just before you break your fast, your body is using about 12% of its fuel is coming from carbohydrate because you've used up a fair bit of the glycogen in your body just lying there in bed. And 70%, nearly 3/4 of the energy, is coming from your fatty acid stores. Not much change in the fraction coming from amino acids. So in just that 12-hour window, you can get a very large change in the overall economy of how your body is running itself. This is from a relatively brief fast. It's one reason why you should always have a little bit of protein with your breakfast. You're starting to shift into another frame. If you fast for a long time, days, essentially none of the energy is coming from carbohydrate anymore. The brain has shifted over to ketone bodies. You remember we talked about how ketone bodies are made from fatty acids. Almost all the energy is coming from fatty acids, either directly as fatty acids or indirectly as ketone bodies, and down to 5% amino acids because the body has been drawing down the stocks of body muscle and is starting to spare them as much as they can. So the body is really capable of an extraordinary spectrum of energy metabolism, and coordinating that so that it's the right thing at the right time without wasted effort and so forth is going to be the job of insulin and its companion in crime, glucagon. In general, insulin as a hormone is sending the signal that fuel is abundant, that we have stuff on hand, store stuff if you can, keep what you've got in the bank account, don't draw something down. And what that translates into in biochemical terms is this. If we look at what insulin does to muscle cells and many other tissues, it's going to increase glucose transport. This is sort of the simplest thing to do. Make sure that you clear the glucose out of the bloodstream. It's going to increase glycogen synthesis. It's not enough that you've got the glucose out of the bloodstream. You want to put it into an insoluble form out of the way. At the same time, it'll be working to suppress the hormones involved in glycogen breakdown. In fat cells, there's a somewhat different spectrum of activities, and this should bring home to you that insulin and glucagon are not just about carbohydrate metabolism, they are also about lipid metabolism. So insulin will stimulate the lipoprotein lipase, which is the one that is used to get fatty acids off of the transport LDL, the transport lipoproteins and into the cell. It'll stimulate acetyl-CoA carboxylase, which you may remember plays a role in fatty acid synthesis, so that if you have a vast excess of glucose, it won't all get stored as glucose. Some of it will get stored as fat, thanks to insulin. At the same time, it's going to suppress fatty acid release, that we have deposits of triacylglycerols in various parts of our body. Insulin will tend to suppress any breakdown of that because the current body status is one of excess energy, not of deficiency. So where does this magical molecule come from? Well, insulin gets its name because it's made by a particular kind of cell in the pancreas, the pancreatic islet cells. And this is an a light micrograph of a section of the pancreas. They've used a stain that highlights the beta cells like this. Now, they're called pancreatic beta cells because they're also alpha islets, and the alpha islets make glucagon. Much of the pancreas's work is involved in making digestive enzymes, but there are these little islets inside of it that make the hormones. One of the cute tricks that happens inside the islet cells is that the actual metabolism of glucose by the islet cells constitutes an intracellular signal to the islet cell as to whether or not to make or not, to release or not insulin. And it exploits this difference in activities. You may remember the enzyme hexokinase, which can add a phosphate to a number of different 6-carbon sugars. Its curve is way over here. It has a high affinity for hexoses and is essentially saturated in pretty much any concentration of blood glucose that you're likely to be alive in. Glucokinase, by contrast, has a lower affinity, and so it has a nearly linear response within the fluctuating range that blood is likely to be in. And so glucokinase activity gives the islet cells a chance to read the concentration of glucose outside the cell. I mentioned before that glucokinase is actually mildly sigmoidal, even though it's a monomer, which is a cute trick and may contribute to how well it does its job. Now, insulin by itself is just a piece of protein. It can do nothing by itself. Like all hormones, it's going to depend on having some kind of signal amplification going on. So insulin is going to circulate in the bloodstream from the pancreas throughout the body, and on cells that have insulin receptors, it will encounter the insulin receptors, which are alpha-beta dimers. So they have two sets of that dimer, of those four peptides all together. The alpha component is a large extracellular component, whereas the beta component is a transmembrane one that spans clear across so that it can interact both with the alpha subunit on the outside of the cell and be on the inside of the cell as well. When insulin binds to the insulin receptor, it induces a change in the conformation in such a way that insulin receptors can phosphorylate themselves. We call this autophosphorylation. In the particular case of the insulin receptor, it has a tyrosine kinase activity that resides in the beta chains, so that one beta chain can phosphorylate its neighbor if insulin is sitting on the outside. There appears to be a role for another component of the system called insulin receptor substrate 1. So it's likely to be a little more complicated than just the simple set of it. But we're going to see this strategy, or if you study this subject in more depth, you'll see this strategy that a lot of the peptide growth factors work through receptors that have a tyrosine kinase activity. Insulin is by no means the only growth-related hormone that's a peptide. There's insulin-related growth factor and human growth factor and a number of other very important growth-related factors that are small proteins or polypeptides. It's also going to be commonly the case that one receptor may have more than one target in order to get the whole job done. I thought I fixed that. I'm sorry. All right. So I've mentioned already that for insulin to have an effect, there has to be a receptor on that cell. And some of the effects that they have, for example, in muscle and in adipose cells, is to increase glucose transport. I already mentioned that. And it accomplishes this trick in kind of a cute way. At the resting state, cells that are going to be able to respond to insulin with a change in glucose transport will have a reservoir of of glucose transporters inside the cell in an inactive state. And the effect of insulin is to mobilize these transporters to the cell membrane, so they're now at the cell surface and can take up more glucose. And so you can have a quite quick response. The machinery is ready to go. You just need to pop it in place like a module. And what that means is that the Vmax for glucose uptake can increase quite quickly in response to insulin. This is much more quickly than if you had to make new channels. If you had to make new channels, you'd have to transcribe something and translate it and process the sugar and so on. So this is a very brisk, quick way to make a response. When the insulin signal is turned off or when other countervailing signals come in, these membrane channels can then be taken back in and they're ready for the next round. So you can reuse the channels more than once. Insulin will also increase fatty acid uptake by increasing lipase activity. So it's going to increase the clearance of fuels from the bloodstream for both sugars and fats. Now, once you have your glucose in the cell, it might be that you're about to run a long-distance race, in which case you can burn it. But you're more likely to find it useful to store that glucose for a later time, such as being asleep overnight. And so to store the glucose, you need to make glycogen. And glycogen is principally going to be modulated at the level of glycogen synthase. And this is sort of a somewhat simple diagram for glycogen synthase activity. Glycogen synthase can respond to some allosteric regulators that will change its activity up or down, but the largest share of glycogen synthase regulation is going to be mediated through insulin and through glucagon. And if we sort of take a look at that, first of all, there's partners of enzymes here. Glycogen metabolism is, like all metabolism, a balance of synthesis and breakdown. And so it wouldn't be very efficient to just regulate synthesis or just regulate breakdown. Insulin is going to modulate both halves of the equation. It's going to increase synthesis and decrease breakdown. And the enzyme that activates or catalyzes glycogen synthesis is glycogen synthase. And the enzyme that catalyzes glycogen breakdown is glycogen phosphorylase, the enzyme that can add a phosphate to glycogen and release glucose 1-phosphate. And so what we can see here is that these two enzymes can be switched on and off by the presence or absence of phosphates on those enzymes. And in the case of glycogen phosphorylase, for instance, it would be activated when it's phosphorylated. When a phosphatase takes that off, it becomes less active. Glycogen synthase in the phosphorylated state is off, and a phosphorylation turns it into the more active state. The dephosphorylation, makes it more active. And so by modulating kinases downstream of the receptor that can target both halves of the equation, insulin can produce an overall accumulation of glucagon when glucose concentrations are high. Life is not always peaches and cream. We are not eating all the time, at least not most of us. And so there are intervals between meals, and there are even these overnight fasts to deal with. And so the body has to have a way to mobilize the various fuels that it's stored in the postprandial state. There has to be some way to get it back out where you can use it. One of the things that's going to happen is that the liver can mobilize its glucose. And there are going to be three major hormones that play a role in that. Glucagon, epinephrine, and norepinephrine. Glucagon gets its name from, agon is as a contest or an urging. Glucagon rouses up glucose and gets it going. You may know epinephrine and norepinephrine is adrenaline and noradrenaline. They're made near the kidneys in the adrenal glands. Glucagon is kind of the fraternal twin of insulin. It comes from the alpha cells of islets. And glucagon acts in a number of ways. Again, it acts on both carbohydrate metabolism and on lipid metabolism. And so glucagon in the liver stimulates the breakdown of glycogen, glycogenolysis. It stimulates gluconeogenesis. So those are both pathways that are going to mobilize sugar from the liver out and are going to turn carbon backbones into sugar for use elsewhere. Glucagon also stimulates adipocyte lipolysis. So the various cells in our body that are principally involved in storing it get given the signal, time to break down those triacylglycerides. So the glucagon is going to be focused more on the liver and the adipocytes. The epinephrine and the norepinephrine are going to be focused more on the muscles, on glycogen breakdown in the muscles. This is one of the reasons why exercise is so very helpful to diabetics, is because it can modulate glucose metabolism through hormones like epinephrine without having to have insulin involved in the equation. Now, glucagon, like insulin, like hormones in general, has to have some kind of a receptor. This is going to be a membrane receptor again. We'll see, I think, later that there are some intracellular ones. But this is a transmembrane one, and it's one of a large family of what are called the 7 transmembrane domain families. So this is a very large family of proteins where there are 7 alpha helices that expand through the membrane to accomplish this. Some other members of that family are the adrenergic receptors, rhodopsin, which is involved with light reception. And in this case, you again have glucagon being the ligand. It binds to the glucagon receptor. It changes the conformation. But this family of membrane receptors are not tyrosine kinases. They're going to get the job done a little differently. What we're going to see is that many receptors need to interact with another set of proteins that are inside the cell in order to transmit the signal. And this group of proteins are called the G proteins. G protein stands for the fact that the protein can bind GTP or GDP. And when GTP is bound, it activates the G protein. Now, like everything, there's going to be ons and offs. So if you have activated the G protein to be released as in the GTP form, at some point it's going to have to hydrolyze the GTP to release it. You now have an alpha GDP, which would have to exchange that GDP for GTP to regenerate the active form. As you might expect, these processes themselves can get accelerated, that there are exchange factors that can accelerate the exchange. There are other proteins that can accelerate the hydrolysis of that last phosphate. An example of a protein in which GTP binding is going to act to alter enzyme activity is in adenylyl cyclase. That if we look at the inactive tetramer, there are 2 subunits that are regulatory and 2 that are catalytic. If this tetrameric protein binds cyclic AMP, in doing so, the regulatory units release the catalytic subunits, and those catalytic subunits now are capable of catalyzing this reaction. And this reaction is one that takes ATP, which we recognize from so many other times in our encounters, and makes this odd kind of a cyclic phosphodiester here, splitting off a PPI. So we call this cyclic AMP or CAMP. And this cyclic AMP is able to then bind to other proteins, such as protein kinase A, because it's bound cyclic AMP, and it will target serines and threonines in turn. Among those would be phosphorylase. So remember, phosphorylase was going to be one of the targets that we wanted to have eventually modulated to regulate carbohydrate metabolism. Always an off switch. The off switch for proteins that are activated through cyclic AMP is a phosphodiesterase that can cleave that cyclic bond into a plain old AMP, and in that case, it's now inactive for activating protein kinase A. What protein kinase A will do depends on what targets for it are in that cell. In the example we just talked about, it was phosphorylase, which would be involved in glycogen metabolism. In adipocytes, it would have targets like the hormone-sensitive lipase, which would be a way to recruit fatty acids. It also has the effect of redistributing the lipase so that it's associated with the fat droplets where it can do more work. This is just giving you a comparison between a couple of these G protein coupled transduction pathways. So we talk about the process of getting from the hormone outside the cell to the final thing that we need to have happen, transduction. And here we're comparing targets ultimately here looking at glycogen synthesis or glycogenolysis or lipolysis. And we can see that in the initial stages, the two pathways are fairly similar in terms of having specific hormones that are going to be bound to specific receptors. Those receptors can activate G proteins. Those G proteins, in turn, can activate adenylyl cyclase. Each adenylyl cyclase can make many molecules of cyclic AMP, which is the second messenger in this case. Those molecules, each one of them will activate a molecule of protein kinase A, which can act on many targets. So we have amplification here and amplification here. But the actual outcome is going to vary with the cell. The protein kinase here is affecting the lipase, and over here is targeting phosphorylase kinase or glycogen synthase to change those activities. So the machinery can be quite similar through this point. The outcome will be quite different because the final targets are quite different. There's an immense amount that could be said about G proteins. G proteins play a role in a lot of other pathways as well. When the human genome was completely sequenced, it turned out that 1% of the human genome genes are G-linked receptors. So it's a very versatile strategy to be used. And they can act in a number of different ways. We just looked at a couple of pathways where they increase the amount of cyclic AMP by activating adenylyl cyclase. They could inhibit cyclic AMP production. They can affect guanylyl cyclase. So in addition to cyclic AMP, you can have cyclic GMP. And they can also target other kinases or lipases altogether. They're also involved in transmitting some of the effects of epinephrine and some of the phosphoinositides as well. So if we were to look at some of these pathways, there's, again, similarities and contrasts. We want to look at, in this case, another family of signaling called phosphoinositide signaling. We've looked, this is to remind you of what we've just been looking at of a G protein that activates adenylyl cyclase and cyclic AMP. In contrast, epinephrine or adrenaline can act on a receptor, activate a G protein, but this G protein is now going to activate a different enzyme which can amplify. It's phospholipase C, and phospholipase C has as its substrate phosphatidylinositol, which it's going to split into two fragments, inositol triphosphate and a diacylglycerol. So adenylyl cyclase makes second messengers. Phospholipase C is going to make second messengers as well. And these then work sort of side by side with each other. The diacylglycerol can activate a protein kinase C in cooperation with a calcium that's been recruited through inositol. Calcium channels can bring in an influx of calcium that can then activate phosphorylase kinase or protein kinase B, and so on. So I'd like to maybe make this a little more concrete or a little more specific or a little more medically motivated by thinking about these things in connection with diabetes and how a disease of metabolism affects the whole organism. And to do that, I'd like to think a bit about diabetes, which is a premier, the most important disease of energy metabolism. Diabetes is called that because people who are manifesting it urinate excessively and there's sugar in the urine. This dog was the dog in which diabetes was experimentally induced by taking out the pancreas and developed diabetes as a consequence. We talk about two types of diabetes, type 1 and type 2. Type 1 diabetes is an insulin-dependent diabetes. We used to call it juvenile diabetes because it used to be the only kind of diabetes that kids would get. And it is essentially an autoimmune disease, that in type 1 diabetes we can see a destruction of the islet cells and the presence of antibodies that are self-reactive. The second type of diabetes is called type 2, or insulin independent. Individuals with type 2 diabetes have measurable amounts of insulin in their blood, sometimes lots of insulin in their blood, but they're not responding to it. We used to call this adult type diabetes. Unfortunately, it's now turning up in kids as well. And it really is a disease of insulin resistance. So type 1 diabetes, you can't make the insulin. In type 2, you can, but you can't tell that you've done that. Either way, whether it's type 1 or type 2, the cells are not functionally getting the signal to take up glucose. It may be there, it may not be there, but they're not responding to it, and therefore there's extra glucose outside the cell. And as a consequence, blood sugar levels rise very high. Since the cells are having a hard time taking up the sugar, they think they're starving. And the cells respond to starvation as any smart cell would respond to starvation. Gluconeogenesis, trying to mobilize fatty acids into the bloodstream so they might be available to be used as a fuel. There are other consequences of diabetes. There's a non-enzymatic glycosylation where if the blood sugar levels are high enough, indefinitely long enough, the glucose just sticks onto proteins and modifies them. And this doesn't just happens by the nature of the sugars. There's no enzymes involved, and it probably contributes to a significant amount of the tissue damage that happens in the course of diabetes. Diabetes is associated with neuropathies, where the nerves get damaged. Sometimes there's neuropathic pain, where the nerve damage gives false signals of harm being done and you can't turn it off. In other kinds of neuropathy, there's numbness, and so it's not uncommon for diabetics to have imperfect sensation in their feet and to then develop foot sores or damage to their feet, which they're not aware of until sometimes it's progressed too far to be treated well. Another common consequence of diabetes is kidney damage. This nonspecific glycosylation of proteins I mentioned can happen in the kidneys as well, and other reasons for damage there. And one of the major causes of kidney failure in the country is due to diabetes. It's also common in diabetics to see vascular and heart disease. This increase in the serum lipids has some of the consequences you might expect from any such increase. Trying to decide whether this is I guess we have time for a couple more slides. we're seeing a vast increase in diabetes because there is presently in America an epidemic of obesity. Obesity is not just a major player in diabetes, and we're going to see some more how it plays a role in that. But it also is significantly associated with heart diseases, with cancer, with damage to the joints. I don't know if any of you know people who've needed to have hips or knees replaced because the joints are just simply wearing out. But those are all things in which an excess of weight over the desirable weight negatively affects the organism as a functioning organism. And it can happen because it doesn't take much of a change to end up with an increase in weight. If you think about it, it takes roughly 350 calories excess, 3,500 excess calories to equal 1 pound of weight. So the difference of a couple of cans of soda a week could be a pound a month. And it really isn't possible to monitor our own eating and our own activities to balance those books as exactly as would have to be done to maintain normal weight by simply thinking about every bit of it. In some people, there's a suggestion that a protein called leptin, which is a peptide hormone relating to fat metabolism, may be off balance. But it's pretty clear that it isn't. The obesity epidemic is not principally due to people who may or may not have primary defects in leptin, because there's been such a change. If you look in 1900, roughly 5% of Americans were obese. If you look at the numbers for 1988, roughly 31% were. So there hasn't been a 6-fold increase in genetic imbalances in leptin in a century. People have traced or attributed some of these changes to a variety of things that are in the environment around us, things that are different now versus 1900. We're awash with advertising for food in general and for junk foods in particular. If you've ever looked at kids' TV shows, you know, the constant flow of the sugar cereals. The availability of processed foods, and we're going to see when we talk about glycemic index what that contributes. The American government subsidizes foods, and when you make things cheaper, people have more of them. That's sort of one of the laws of economics. America has an automobile culture. In general, Americans drive a lot of places, and you can show that in 2 of the American cities where people walk a whole lot, which is New York and Washington, D.C., there's a lower obesity rate. And then the shift from 1900 to today, from active sports to spectator sports. In 1900, there was no TV. If you wanted baseball, you played baseball. Nowadays, you watch someone do it much better than you could ever do it yourself. So I'll see you all on Wednesday.